Hey guys, hope everybody's doing well. This is going to be a pretty short PowerPoint, I believe. We're going to talk about 6.4, which is translation. But at this point, you should be somewhat familiar with translation because we've talked about it a little bit already when we talked about transcription and the central dogma and all of that. So once again, our enduring understanding is that heritable information provides for the continuity of life. You continue to hear that again and again. Our first learning objective here is to describe how the phenotype of an organism is determined by its genotype. So what we mean here is the genotype is the DNA. The phenotype is the physical characteristic of the organism. So how do we go from DNA to physical traits? And that is, by definition, what translation is. We're essentially translating our DNA into proteins that determine our phenotype. So the first piece of essential knowledge here is that translation of the messenger RNA that we created during transcription, translating the messenger RNA to generate a polypeptide occurs on ribosomes. So we should know that already. Creating a protein happens on ribosomes. But it happens on ribosomes that are present in both the cytoplasm of the prokaryotic and eukaryotic cells and on the rough endoplasmic reticulum of eukaryotic cells. So if you think back to the unit on cells, you might remember that ribosomes can be free and floating in the cytoplasm, or they can be attached to the rough ER. These little green circles represent ribosomes. And these little green circles here also represent ribosomes. So the first piece of essential knowledge for translation is that the free-floating ribosomes and the ribosomes attached to the rough ER both perform translation. Which one does the messenger RNA go to is usually determined by what the protein will eventually become. If the protein needs to be modified and sent to the cell membrane to leave the cell, then it's probably going to be produced on a ribosome that's attached to the rough ER, because then it can enter the rough ER and go through the steps of rough ER to Golgi apparatus to cell membrane to out of the cell. If it's a protein that will be used inside the cell, then it can probably be produced on a ribosome that's freely floating in the cytoplasm. So the process is the same. The messenger RNA comes, it hooks onto the ribosome reads the code and spits out a protein. Same thing here. The ribosome's attached to the rough ER. The messenger RNA gets read by the ribosome, spits out the protein, and that protein that's attached to the rough ER now enters the rough ER and continues to be modified as it travels through the rough ER and the Golgi apparatus. If this is a prokaryotic cell, we don't have endoplasmic reticulum. So obviously the only scenario here is the ribosomes in the cytoplasm that can translate the messenger RNA code. The second piece of essential knowledge relates to what I just talked about. In prokaryotic organisms, so this is a prokaryote here, notice there is a circular chromosome of DNA, and you have a couple ribosomes here freely floating in the cytoplasm, but in a prokaryote, there's no other organelle or organelles. In prokaryotic organisms, translation of the messenger RNA molecule occurs while it is being transcribed. So, as it's here in this prokaryote, as it's transcribed, ribosomes come and start translating it before it's even finished being transcribed. So pause the video in a second. Think about why that can happen in a prokaryote, but it cannot happen in a eukaryote. So why can we transcribe and translate the messenger RNA at the same time in a prokaryote and not in a eukaryote? So pause, think about it, write it down. And the answer is because in a eukaryote, we have a nucleus. Transcription happens in the nucleus. So we transcribe the entire gene onto a messenger RNA molecule. We have the processing that takes place with the poly A tail and the cap on the front of the protein, and the removal of the introns, and then the exons are released from the nucleus, and then once they enter the cytoplasm, the ribosomes are able to translate the message. So the reason prokaryotes can do both at the same time is because there's no nucleus, so as soon as that messenger RNA starts developing, it can be read and translated. The next piece of essential knowledge is that translation involves energy and many essential steps, including initiation, elongation, and termination. So transcription actually has the same 3 steps: initiation, elongation, and termination. But when we're dealing with translation, when we did the biozones on Tuesday, we talked about the start codon, which, if you can see here, is AUG. And we talked about the stop codons, one of which is UAG here. Initiation is the ribosome forms. So you've got the large ribosomal subunit and the small ribosomal subunit. They form a ribosome. And the transfer RNA carrying the UAC anticodon comes, hooks on. Now recognize this is the messenger RNA. It hooks on at the start codon, and it brings its first amino acid with it, which is methionine. That's initiation, when that first transfer RNA molecule binds, carrying methionine and matching up with the start codon of the mRNA. Elongation deals with the 3 codons following the start codon. Another transfer RNA would bring its amino acid. They would move down the chain. This spot would open up again. The one in this spot would leave. Another one would come. It would start building this long polypeptide chain, and it would continue moving down the line, building this polypeptide chain as it goes. So that's elongation. And then finally, termination. There's a release factor that will read the stop codon. Once the stop codon forms, the release factor binds. It allows this polypeptide chain to detach. This polypeptide chain will now fold up into a protein and either stay in the cytoplasm or enter the rough ER, where it can be further folded and modified. And this constitutes termination. And at this point, the messenger RNA gets broken down. The ribosome breaks down into its smaller subunits. The transfer RNA molecules are free, and all of these pieces can then be reused the next time we need to translate RNA into a protein. All right, so the last slide here is sort of a recap of everything we talked about. Oh, sorry, not the last slide, but this is a recap. Features of translation. It's initiated when ribosomal RNA in the ribosome interacts with the messenger RNA at the start codon, so that's initiation. The sequence of nucleotides on the messenger RNA are read in triplets, which are called codons. Each codon encodes a specific amino acid, which can be deduced using the genetic code chart that we did in the biozone the other day. Many amino acids are encoded by more than one codon. Remember, we talked about that and how that helps prevent detrimental mutations. We talked about how all living organisms use the same genetic code, which is evidence for the common ancestry of all living organisms. That's very important. And that's a good answer for a free response question. If it asks about what this means, it means that we're all, a good answer is that the same DNA suggests unity of life and one common ancestor and common ancestry among all living organisms. Transfer RNA brings the correct amino acid to the correct place specified by the codon on the messenger RNA. Once that transfer RNA binds, the amino acid that it's carrying is transferred to the growing polypeptide chain. And then the process continues along the messenger RNA until a stop codon is reached. And at this point, the process terminates by releasing the newly synthesized polypeptide, and the protein is then able to fold up and form the protein. If you're not an auditory learner and you're a visual learner, I recommend just watching this Amoeba Sisters video real quick. You can pause it or just Google Amoeba Sisters protein synthesis. Spells it out really nicely and gives you a good visual of this process. All right, last slide now. We're not going to talk too much about viruses right now. I'm thinking about going into this a little bit after the exam, but I just want to mention it because it's the last piece of essential knowledge for Unit 6.4. We call these retroviruses. Lots of viruses don't carry DNA, they carry RNA. So these retroviruses are special cases, and they have a different flow of information. Instead of going from DNA to RNA to protein, they go from RNA, and they use this enzyme called reverse transcriptase to essentially transcribe that RNA and turn it into a DNA using the same method as before, where it uses the complementary base pairing and basically forms DNA from that RNA molecule. And then that DNA gets integrated into the host cell DNA, and then the virus can use the host cell to make more pieces of the virus using its DNA, and then essentially release those viruses into the body to infect new cells. Very interesting process, but we don't have a ton of time to get into the ins and outs of it. I don't expect that to be on the exam, but I just wanted to mention it in case. So that's it for 6.4. Today, you should finish your transcription and translation guided tutorial. I want you to do tutorial number 2, the genetic code, and tutorial number 3, from gene to protein. Take all the quizzes, and once you finish this, please upload it to the portal, and make sure your quizzes, check your quizzes, make sure they're complete. Send me a message if you have any questions, and have a good night. All right, see you guys soon. Bye.